As part of the custom Frontiers in Organoid Medicine Symposium held in 2018 at Cincinnati Children's Hospital, prominent organoid researchers participated in a discussion panel exploring the challenges facing organoid technologies as they move from the lab to the clinic. Our panelists are Dr. Cindy Osborne, CMC reviewer at the FDA, Dr. Kevin Demore, Chief Scientific Officer at ViaCyte, Dr. Philip Kramer, Senior Scientist at STEMCELL Technologies, Dr. Michael Helmer, Surgical Director of Intestinal Rehabilitation at Cincinnati Children's, and Dr. Kazutoshi Takahashi, Staff Scientist in the lab of Dr. Shinya Yamanaka. Now, let's turn it over to moderator James Wells as he kicks off this discussion.
Basically, the idea is to just have a few guidelines, ask the panelists some ideas about what the real hurdles are to generate meaningful new therapeutics or diagnostics that are based on pluripotent stem cells. And again, the basic areas that I thought might be good to cover, and again, people can really discuss what they want, are uses of stem cells and organoids for disease modeling and diagnostics. So, for example, can we use an iPS-derived organoid as a diagnostic, broadly speaking, to, again, predict hereditable forms of cancer or other things? And, of course, drug testing, drug efficacy. Some of this is already being done in Europe, where they're using organoids to pre-screen drugs for efficacy. And, of course, transplantation-based therapies. We'd also like to potentially talk about how do you transition basic discoveries from the bench to startup to actual pharma-based screening platforms and therapies. Maybe try to get some discussions on how you might have industry, academic, collaborative partnerships. And then hopefully Cindy can guide us a bit on regulatory issues. Again, how does the FDA handle anything from using organoids as a new diagnostic test to using organoids or stem cell derivatives for therapeutic transplants? So what I thought we might start with is just a brief thought that the panelists might have on what they think the biggest hurdles are to translating pluripotent stem cell derived tissues and cells to some therapeutic use. What are the biggest hurdles that you've observed in your own little microcosm? So why don't we start with Cindy?
I guess for me, just reviewing files from sponsor, the main data that we want to see is consistency, like differentiation of IPS to the product. And I feel, and you can tell me if it's a hard thing to get consistent, identity, consistent proportion, and and do it multiple times in multiple donor. So those are the things that we normally look for. And then in terms of safety, just a good preclinical data. And I know it's hard to get a good model organism to do or a disease model. And so those are the two that I feel like that's the biggest stumbling block every time I review these kind of files, is to be able to manufacture them consistently and to be able to show us that it's safe in preclinical data.
In general, do you think that the field is, the FDA is okay with small animal models, mouse and rat, or under what circumstances would you really find a larger animal model absolutely necessary?
Actually, we don't encourage you to use a lot of animal. There is the rule that, the minimum use of animal, we really support that. So if you have just a good in vitro and a good small animal like mouse, it's necessary. But I understand some of the transplantation, it's hard to show with just mouse. So, but we really encourage you to use the least amount of animal, really.
So something like neural transplants, I know are in the pipeline. Something like that wouldn't require primate or something like that?
Depends. Yeah, it really depends on how good your product is. But there is no requirement that I know of that you need to do primate.
Great. Well, we'll come back to that, I'm sure. Kevin?
So I guess my perspective, the biggest challenge is delivery of cells to human patients and how that's way more challenging than these suitable preclinical models, mice and rats. So sort of, I agree with what Cindy said. There is first the prerequisites to even get into clinical study of consistency, CMC, characterization, preclinical studies. But even when all of that is done and looks beautiful, there's a new challenge, which is those models don't really predict cell survival and delivery in the clinical setting. So I don't know that there's much we can do about that, but get into clinical studies and begin exploring it. That's certainly where Biocide has been for the last 4 years and continues to be. And it's just something I think we'll all have to acknowledge, that these products will be optimized in clinical research, not just clinical development. And that's certainly, I think, the biggest challenge we're facing right now.
And of course, with what you guys do in terms of delivering cells in a device, that's perfectly reasonable. But if you want to inject differentiated neurons into a human brain, there's no getting them back. So one might imagine that the point you're making, which I think is one that everyone's facing, that the small animal models just really aren't good models for a lot of things regarding human cell transplants. It'll be interesting to see how that gets received moving forward.
And, I think either imaging or biopsying, thinking proactively about how you'll do that in the clinical setting, so you don't just deliver cells and look for clinical endpoints 6 months, 1 year, 2 years out, but really thinking, can I look at stuff 1 week in, 1 month in after delivery, and learn something at least about how it's going without just, having a blind eye and looking way downstream for a clinical effect.
Okay, so yeah, I also identify like 3 major hurdles, which I believe could be taking a while to solve. Which is, one thing is, if we think about like autologous cell therapy, bringing their own patient cells, reprogramming them, generating iPS cells, maybe correcting a disease depending on the question, and then bringing them back into the patient, is obviously it takes a tremendous time to actually do that. Is that applicable? Maybe for spinal cord injury, doesn't, most likely not. Then also the costs which are affiliated to that are tremendously high. So is that suitable? And then the third one is potentially also thinking about alternative ways. Cell source, the cell source. I know that we are in this panel discussion focusing on iPS cells, but maybe adult stem cells would qualify as an alternative cell source. And then if we go the way around the iPS lines, then obviously we have to take tremendous care about the safety. Do we acquire any kind of mutations throughout the maintenance and reprogramming, as well as genetic correction using CRISPR-Cas9 or other genome editing tools? So these are, I think, the things we are going to work in the next decades on. But on the other hand, we can already see how the field has moved forward in the past and have actually overcome major hurdles of, for instance, coming up with very defined and even compliance of the cell culture medium, or the extracellular matrix proteins we are using to culture cells in a clinical grade. So,
Kevin, can you tell us how much money did it take to go from the basic science that you guys were doing to make these cells to where you are now? I mean, has anyone done I'm sure you guys know it. Are you allowed to tell us? And I guess to follow that up, I mean, how hard is it going to be for companies to recoup that cost in a way that it's a therapy that a lot of people can afford, as getting to the question?
So I think I can vaguely tell you. So, ViaCyte joined forces with Betalogics, which was a wholly owned J&J subsidiary. So those two groups really have led this field as far as companies working on the space of pluripotent derived pancreatic cells for islet replacement. And on both sides of that equation, total money spent is over 200 million each. So we're now pushing on about 500 million and have yet to show clinical proof of concept in phase 2 style trial. I will say that maybe different than many fields, we did a lot of the earlier stage stuff in the company environment where that would more typically have been done in an academic setting, at whatever pace, but at a much reduced expenditure because you didn't have all the carrying costs of having, companies of dozens to even 50 or 60 people working on it. But the expensive stuff is even yet to come for us. So,
well, I guess isn't the average cost of a drug, isn't it $2 billion, something like that? So I guess you guys are actually under budget.
That's scary.
So I think I'm going to echo some of what was said, but in a little more context with what Cindy said. I think most of us are passionate and we have great labs. And research is very different in my eye than creating tissue that's going to go into a patient of mine. And so QI and understanding the steps and picking out what's going to be patterned to become intestine, colon, stomach, and is that every time is not really of the interest of the NIH, but it is desperately of interest in anyone that wants to translate this to human diseases. So you have to know what you're doing. You have to be able to do it in a GMP way and not lose it. And then more importantly, we have to understand, as we've talked about, why some IPS lines and others actually do better than others. So we had a talk on that yesterday. So we have lots of clones. So when we make these clones, we make a lot, and we keep picking till we find the ones we like. But as was mentioned, if this is a patient that's sick, we don't have time to do that. So efficiency, throughput is all things that have to work with industry to be able to remove what's the eyes and the hands in my lab, that I call it gardening roses. So the people in my lab, I just think, are phenomenally talented. Their eyes know what they're doing, but they can't always say it. And learning how to say it and reproduce it is a challenge that we try to tackle. Second, I think it needs to be understood that the animal models are immunosuppressed and humans are not. And so the immune system plays a huge role in human diseases, and how to do that is a challenge without putting it in a human. Creating IPS lines from primates and stuff, it might be an alternative, but we're not studying human diseases. So we have to recognize the hurdles. The delivery systems, I think, is an issue, but the bigger issue is the fact that you have to realize that diseases like end-stage liver failure, colitis, and stuff is a hostile environment. And what we do in the lab is place things into an exposed niche that probably is healthy and isn't chronically diseased. And so we have to understand the limitations of what we're doing. And I think the last comment is the biggest one, is that, like, for ViaCyte to do this on the outside was great, but institutions like ours need a space that interfaces between what drives us daily in our labs and what gets things out. We have resources and knowledge here. They have resources and knowledge here. If we can't figure out how to work within the same spaces, the delivery of what we dream of is just delayed insurmountable. So I think it's our charge in the room to find best ways to integrate, work together. And that's part of why when we come together, we've discuss this. So hopefully other centers will agree that having open places for collaboration that may not just necessarily be in your lab but are in the institution, I think are the future of trying to take diseases out and doing it in a timely and an economical way.
I think that speaks to one of the biggest hurdles from an academician standpoint is this black box where you have a great fundamental discovery, but turning it into a commercializable product involves a whole different set of parameters. As you said, potentially collaboration between industry and academia in a safe space, in a sort of incubator type space might be one way to do it, but I still find that's probably one of the biggest challenges for getting all this great basic research into an applied clinic.
So one last comment, it's team science is really, I think, paramount to making this happen. And it has to have a vision that has a return on investment which puts the patient first. So, for example, there clearly may be economical ways to take your stuff to market that may actually have a return, but biologically is not what's practical and right for patients. So just because you can get a 3 to 5 year budget sheet that looks like you can raise money and bring something to market, if it doesn't really make sense biologically, should you do it? And I feel that part of our voices in working early with partnerships is making sure that the companies take the pathway that gets it to market the fastest that's going to work. And so we can talk about examples of that, but I can give you several where it would make very clear sense we could do a product. But my own sense is that it's probably not going to be the right thing. And I think the FDA will want us to have markets. But everyone has to remember, we don't patent a human developing tissue. So God gets the patent on that. So we tend to look for ways that we bring things to the product that we can get IP and licensing and that on it. But we still, at the end of the day, when I'm gone, the only thing I care about is the human lives that we change. So I think the mission is to make sure that everything you do in this room will affect humans long after you're gone and who will never know your name. So I think that should always be one of your missions and your keys.
So now I understand the making organoid is still very challenging. So, but in terms of clinical use of iPS-derived organoid, so one of important technical challenge is making freeze stock. So, so I learned how to make cell bank for clinical use when I was in Japan. So usually we should make master stock and then working stock. And after making freeze stocks, so we should evaluate, we should check the stocks. So if they are safe or if they are high quality. So every time we need viral contamination check and also if there is a genomic mutation or not. So, sorry, I don't know if making freeze stock is necessary every time, but if we can make this, that's very great advantage, I think.
Jim, that's I'll follow up on a question for you, Kazu. It gets to the safety issue. So I guess one of the concerns is ultimately when you transplant undifferentiated cells or teratomal, it's always an issue. So in your experience with all the work you've done in iPS cells, I mean, what's your view on this, that some cell lines are going to be better in terms of making a transplantable product? Or do we have to think about, people are looking at making a suicide cassette so you can kill undifferentiated cells? What are your thoughts on some of those?
Yes, actually, one paper suggested less than 20 cells of undifferentiated cells can make teratoma after transplantation. The residual undifferentiated cells very risky. But other than undifferentiated cells, so there is a possibility of de-differentiation of differentiated cells. So it's possible, and it's also very risky. So yeah, so to confirm this, so we need to evaluate the working master and working stock.
I sort of have a question for everybody based on the last comment, which is as a physician scientist, I think of myself in the lab. Based on Michael's comment, like in the lab, I think this is really cool. We should think about how to translate this. And then as a physician, I have a substantially different bar for what I'm willing to put into my patients. If someone comes to me and asks me to enroll a sick patient into a clinical trial, especially when you take into account the sort of risks of potentially a small number of cells. So one of the things that I've been trying to figure out, and I'm really interested in people's comments on, is how much science is enough before we translate, and how do we translate in an efficient and economical way at the same time as we're developing things?
So as a clinician, I am posed with challenges that have few to no solutions with dismal to no hope. So I honestly feel we in an environment of developing children and stuff that have no real hope, and that you're trying to figure out a baby with no trachea, and you basically have no way to get an airway, and you can put a decellularized graft and it's going to blow out in 2 months. We know that. I think it's a contact sport, and you have to be realistic about the advantages. To put tissue into an otherwise healthy patient to make them a little bit better is not your first patient. You have to select the right diseases and the right situations, and you have to have an informed process with families that understand what you're doing so that it moves forward. So it's picking the right situations and the right clinical scenarios that I think are very key. And we have plenty of them. And so I think you totally are right. If you wouldn't do it on your child, don't do it on your subject.
I would like to add something to that. So I also believe there's a huge opportunity for academia and potentially biotech to move things forward. So myself as a now working for a biotech company, I see these great technologies being developed in academic labs, but then really facilitating them to move them into like a translational trial or study. I see like it would be really great, a great opportunity to cut down time in respect to developing compliant media, developing methods and protocols, manufacturing procedures, to actually start to collaborate early and partner up early, because that can really help to, for instance, identify components for the products which are compatible with any kind of CGMP or elsewhere, and then do a lot of these medium protocol optimization and compatibility, everything in parallel rather than one after the other.
Now, that's an excellent point for expediting the translation, is doing it in partnership as early as you can in a GMP manner. And this gets to Kazi's point of introducing quality control metrics alleviates many of the issues and introducing those early in the process, first of all, is just good science, but second of it all, really allows you to document robustness of the procedure and translatability.
Exactly, and this brings us back to once you go into the patient, you feel comfortable that everything you could do in advance was being done.
Okay, I want to continue opening this up for questions. So
one point that's linked to picking the good clones, and that's coming up in the discussion for like international stem cell banking initiative, is that one of the quality control that will be done is exome sequencing. That has two downfalls. First is you will know who is the donor of the cells you will inject, which, when you think about brain cells, can come as a new ethical question. Secondly, you will know the genomes of people. So if you want to, have cells from someone, you might be better want cells from someone that's less risk to develop diabetes if you want pancreatic cells and so on. So that's opening up a whole new perspective on cell therapy, because it's actually genome therapy, like using someone's genome to fix another person's genome. So how do you integrate that in your thinking? How do you integrate, like, telling the people that are donating samples what is really going to happen down the road if the cells are used for cell therapy?
I've always thought the best cells for a patient are their own. So IPS that matches has all the benefit, but there's clearly going to be situations where that's not. Have the groups taking HLA factors and making off the shelf available nerves to put in. That may work. It also may be that those HLA factors are how we sense and respond and heal in a toxic environment. And so those are all things to end up happening. I think how the genome and identifying patients is going to move forward is a big question that is not just limited to this space. As we start to do more sequencing, in fact, in this institution, it wouldn't be unrealistic for us to be doing 23andMe on every patient because it gives you enough information to move forward. But how it could be used in detrimental ways is for society to start to tackle. I think our charge is to take our science and to apply it and let the social networks that govern us give us the best ways to protect the information and to inform patients of the risks that exist. And then at the end of the day, when you go put your head on the pillow, just be confident that you did what was best, and that the world is full of bad, and that doesn't mean that you should be afraid to do good.
But I see it as, stem cell derived cell and tissue banks, I don't see as being any different than blood banks or organ donation centers, in that, these can be treated as donations, without identifiers and whatnot. I realize that's a whole separate discussion that eventually we can identify everyone by sequence. But at the moment, I think developing tissue banks could fall under the same regulatory umbrella as a blood bank or an organ donation center.
Thank you. It's all kind of connected questions. So tissue banking, iPS banking is a great idea. But I think before that, we need to make a international consensus, because someone used Matrigel, someone used laminin. Even the laminin has 511, someone has 411, 211, and we compete each other. And then one company sells MTZA, one company sells STEMFET. One is better, one is better, some cheaper. Can we have like international standard? Can we just generate from this group? It's an international group here, and it's companies here. Can we make one collaborative company or a joint commission to make it standard and tell the world that this is the standard, this should be the standard, and without this standard, your research are not in standard. Can we convey those messages to the world? Or one thing is, can we stop competing? One thing.
We're human.
Because one company makes better medium, one company makes cheaper, but it's reasonable. And Matrix is very different. I mean, come on, Matrigel? Do we use it for human? So can we have that one big body to convey the message, standardize, and make it more available for researchers too?
You sound like John Lennon.
Does anyone on the
Not a
panel
bad
want to
sign.
tackle
No, it's
that
a
one?
very good. It's a good point. But how do you enforce it, and how do you know what the best is? I mean, I would argue when everyone says standardize everything across the consortium, we continue to evolve. I mean, how do you evolve together? The way you evolve together is team science, and you are willing to share things across communities in a timely, real manner. But I don't know how you enforce what you're going to do.
We are supposed to be the best organoid center.
Huh?
We are the best organoid center here. So if we say this is good, this will be the best.
Well, I'll give you an
Maybe.
example. Every person in my lab generates intestinal organoids slightly differently. So if I can't enforce it in my lab, it's going to be pretty hard to do it uniformly. But I think the point is that we're constantly getting better. So we don't want to lock into a subpar approach to get there. But once you go to clinic, obviously, and Cindy and Kevin can speak to this, you do have to lock into one method that's been quality controlled. And even though you might have developed subsequently a better method, you still have to stick with that one method until you go through and show all the quality control metrics for your new method. So, Ed, you want to add anything to that?
Yeah, I don't know that even if there's a gold standard, say this is the best media for ES cell growth, it's not always the cheapest. I can tell you right now we're using one of the cheaper, and ES cells is already the most expensive part of our cost of goods from a reagent basis. So if you told me I had to use laminin and the latest and greatest media, I would tell you no way, because ours is totally sufficient for our needs. Might not be sufficient for everyone's needs. It might not work on 9 different pluripotent lines. We only need one pluripotent line. We don't need any more than one line. So if our cheap media works for our one line, that's what we're going to use. We shouldn't be forced into using something that throws a monkey wrench in our process that was locked 8 years ago, right? We haven't changed a single reagent since 2010. And with that comes the power of knowing, 300 times in a row this process works, right? Because we haven't been forced to evolve with the field in our use of reagents and methodologies.
Sorry, I just wanted to say, I wanted to add a little bit to your statement that it's better if you lock your process early. And we normally don't advise you to change anything because then you'll have to do comparability testing. But we've seen sponsors do it. It's a pain, but if you think it's going to make your product better, come to us and we'll help you go through that comparability study.
Rafi?
So I want to offer an observation and then ask a question. So the observation is, and it was echoed in the comments you heard, is that there is a massive collection of talent in this meeting, and it's up to you how to leverage that to create a smooth sailing forward in terms of fair play principles and coming together and thinking how, without losing the competitive edge, without losing sight of what makes us all human, still work in a civil manner and in a collaborative manner, projecting what I've heard so far to make the progress forward go even more smoothly. But now to the question. So it makes perfect sense to me to address a disease like diabetes that has a very large market and a lot of need, and it can be addressed with an encapsulated product, and we heard several versions of that. Where is the bottom? What is beneath the commercial and regulatory logic to pursue for this type of therapies? We heard of 2 patients very many decades apart that can be helped by this technology, but the cost must be staggering. So what are your thoughts? What's the market slice that needs to be there for a company to be interested in taking care of it?
I guess I can talk about that. We've been working on this since the company was founded in 2000, so 18 years. The reason it's been resourced as well as it has been for that long, being yet so far away from the clinic and application, is just what you say. It makes so much sense to treat type 1 diabetes this way. So even if you said it's going to take you 20 years to get there and $1 billion to do it, there are people out there that would say that would easily be worth it. There's $18 billion of insulin sold every year or something like that, right? And I think front runners like these are necessary to lower the bar ultimately, to lay regulatory framework, CMC framework. The science, is already doing a great job of laying framework on a ton of other applications for pluripotent cells. And with some successes, I think that bar can get even lower, creating, financial resources for reinvestment, as well as a more, straightforward framework on how to take a therapy more efficiently through a process and into commercialization to actually help. help people. I'm sure if you ask Genentech or something, could you see making XYZ protein to treat, 42 people around the world, they'd say, no way in 1988, right? You're only going to focus on the biggest and the brightest, yet then there's companies like Genzyme that come along and do enzyme replacement for literally, few hundreds of patients, right? So it's already happened in other therapeutic arenas. I think it eventually will also happen in cell therapies and possibly even pluripotent-derived cell therapies.
And doesn't it speak towards team science? Because although you have 20 orphan diseases, there's commonality among all of them in the processes that have to occur. And whereas ViaCyte had to really do it in a silo because you were a forefront runner, it doesn't make sense to me that you wouldn't integrate your science to do technology for a brain or for a heart or for a bone or for a gut, where a lot of that just, the similarities are probably more than the differences, and especially getting things started. So I think that's where the community has to come together, and that's where the things like we're doing here hopefully get groups together that would not otherwise study things because the biology might be different, but the starting points, the structures, and the questions, I don't think are that different.
Do we have any other questions out in the audience?
A question or a comment. Really, it gets to the issue of we have to be careful, because if some of the first inpatient trials go wrong, that's really going to set the field back. I've returned to Mike's point about the trachea transplant, right? So we've all seen where some of the early problems from Karolinska, where they tried to transplant trachea, these patients had died basically, and now, even though I think yesterday we heard probably about some real viable options for transplanting artificial trachea made from cells, now I think that's before the public are going to be comfortable with that, we have a lot of problems. And so, it gets back to people just squirting in stem cells to try to fix something. I think we have to be very careful as a field so that we don't set ourselves back with some early missteps. I don't know I guess there's not much to comment on, but I think that's something to think about.
And I think the gene therapy field felt that acutely, where the first clinical trials ended up killing some people, set them back 20 years. So I think that's an incredibly important point.
And this should all be a risk-based, set of processes. So someone was bringing up, I think Mike and someone from the audience, if you had a kid that has very low chances of having survive or having any kind of quality of life, there's a different risk-benefit ratio there than there is for type 1 diabetes, where people can live 50 years reasonably well, like a lot of effort on their back, but reasonably well. So the standard of care is so high, the tolerance for risk is very low in that setting. So you got to take each one on its own. And yes, you have to be safe overall, but look at CAR T therapies. Those were killing people along the way, but the efficacy was compelling, and that field clearly has suffered not from people dying during clinical trials, unlike what gene therapy did. So it's all about who you're treating and what their prognosis is and how you go about it.
Also, one more thing to add to Aaron's comment is that, for instance, I have the feeling that the perception in the public is also, if they hear stem cell therapy, they immediately think about taking those stem cells and using them for cell replacement therapies or cell transplantation. But I think we also should start maybe more emphasizing that, again, what we heard today in Jim's talk, using stem cells, disease modeling, for organoid-derived drug testing, and putting this out in the public and this is where we already are. And then it's just a question of time once we move forward into more cell therapies.
And I want to echo that using stem cell and organoids to pre-screen drugs for efficacy is happening today. In fact, in the Netherlands, insurance companies actually pay for it, which I don't think will ever happen here, but it should be. We should appreciate the accomplishments that have already happened and the impacts they can have on patients long before we start ramping up for cell replacements.
I mean, I would like to echo the comment about CAR T-cell therapy. I had the pleasure of taking care of some of those patients during my training, during the active clinical trials aspect of that therapy, and they were really sick and some of them died. Some of them did not do well. But I think that the lessons that I learned from that, and some of my colleagues, both physician scientists and physicians, learned from that, were they had the benefit of choosing a disease that was a disease without a cure. These were patients that had failed multiple therapies for their hematological malignancy and were not doing well. They engaged with partners in multiple different subspecialties outside of oncology to try and develop a coordinated care system to take care of the patients. So the intensive care physicians and the ID physicians and everybody else who was part of the process of taking care of these patients was bought in because they all knew that it was for the benefit of the patients. And they took advantage of really good press about their early successes. So there's a young woman who nearly died from an early CAR T therapy who now is alive 5 years after an incurable malignancy at the Children's Hospital of Philadelphia because of the therapy, and they utilized that to deliver to people who were not as involved in the science and didn't understand the nature of the discoveries, why this might matter. And I think that those are some lessons that maybe as a community we can learn from in partnering with each other, but also partnering with people outside of this room to help them understand why these things are important and how we're choosing the patients so that if we have challenges, we understand that those challenges are part of a process that's directed towards patient-centered values.
Great. I think if there's no more questions, please thank our panelists for a very interesting discussion.